right, so why do we do sexual reproduction? The answer is actually not clear. We infer, because it is the prevalent mode of reproduction in all eukaryotes, essentially, except for a few that have lost the ability to reproduce sexually, that it somehow pays for the immense cost of sexual reproduction. And when we think about the cost, that comes in several ways. The most obvious is that if you need two parents to make progeny, then it makes reproduction much less efficient than if a single organism can reproduce itself. So there must be some payoff for this. We obviously have a lot invested in sexual reproduction, both individually and as a species, and it requires a lot of biological innovation. Both the whole process of meiosis and fertilization are very complex, and the organisms themselves have to actually get together and mate. And we're not going to get into any of that, but it's a whole fascinating area of biology. So meiosis is a very, we infer that it has very favorable impact on the reproduction and the genetic diversity of progeny, but it also creates a lot of problems that organisms have to solve. It creates sort of a battlefield for selfish DNA elements and other kinds of shenanigans, because in order to get transmitted from parent to progeny, a selfish DNA element like a transposon or other kinds of mobile elements want to leverage what happens in meiosis to their own advantage. So that also drives rapid evolution of meiosis. And yet, as I said, all of these complications seem to not outweigh the advantages of sexual reproduction. So you've already learned about meiosis in a sense by learning genetics. And so today I'm going to focus on the chromosome gymnastics that enable recombination between parental chromosomes to happen and enable proper segregation of chromosomes during meiosis. So we talked about mitosis on Wednesday, and meiosis, as I said, is sort of like mitosis on steroids, in the sense that all the same things have to happen that happen during mitosis, except in addition, there are many additional layers of chromosome organization and recombination that have to happen. So this is just to illustrate something that the book illustrates, in my opinion, in a more clumsy way, which is that the whole cycle of sexual reproduction involves going from diploid to haploid and back again. So we and all eukaryotes are generally diploid, meaning we get a set of chromosomes from our father and a set of chromosomes from our mother. So we have two sets of chromosomes in most of our cells, but we also have a specialized germline that produces these cells that we call gametes, like sperm or eggs or pollen, that are haploid, and those are essential for reproduction. If we didn't do that, then the number of chromosomes would double each generation as sperm and egg fused. So they have to be haploid. This is basically the book's version of the slide I just showed you, but I like mine better, so I just put this in here for reference. So I mentioned that in animals, in us, we have sort of dedicated tissue that does meiosis, and that tissue is called the germline. Sometimes it's written as one word, and it's set aside early in development. I showed you a worm embryo with that early cell that had the P granules around it. That was the cell that's going to give rise to all of the reproductive tissue in that animal. And similarly, when a new human embryo arises very early in development, there are cells that are set apart to become the germline. And the rest of the animal is collectively known just as the soma. That's fairly unique to animals. Plants, as you know, if you've ever done any gardening, don't have a sharp distinction between germline and soma. That's why you can take a cutting from a plant and plant it, and it will produce eventually flowers or other reproductive structures. Okay, so the way that meiosis happens is that there is a single round of chromosome duplication or replication, or S phase. But unlike mitosis, where you have just a single division, there are two meiotic divisions. The first division usually, not always, separates the homologous chromosomes, the copy that your mom gave you and the copy that your father gave you get separated, except that they're scrambled before they get separated. And so they're not purely mom or purely dad anymore. And in the second division, the second meiotic division is actually a lot like a mitosis. The sisters separate very much like they do in mitosis, so they're fairly similar. So just to go through kind of an illustration of how this all works. This is just a schema, like a diagram of a nucleus that has just two pairs of chromosomes, so just for simplicity. And so there's this yellowish pair and there's a bluish pair, and they want to separate from each other. In order for that to happen, they first replicate, as I said, so they go through an S phase. Some people consider that to be part of meiosis. Some people consider it to be a prelude to meiosis. I don't really care. And following that S phase, there's a major change in chromosome organization, which I'll tell you something about. The chromosomes become highly elongated. They have to somehow find and pair with their partner. So here they've already done that. They also undergo a process called synapsis that I'll explain a little bit more, but they assemble this stuff between them that helps glue them together. They also undergo programmed double-strand breaks, which Gary talked about a little bit. They're made by an enzyme called SPO11, and they're highly regulated so that every chromosome gets some breaks, but not too many. And then those breaks get repaired, and a subset of them get repaired in a way that gives this special kind of structure called a crossover or a chiasma between the homologous chromosomes. And that holds the, sorry, I'm moving my mouse around. I have other things going on my screen. So all of this stuff from pretty much the end of the S phase through crossing over and then condensation of the chromosomes is considered to be meiotic prophase. It's a little bit of a misnomer because they're not really in prophase like they are in mitosis. It's more like a extended G2 phase, but it's a specialized meiotic prophase. At the end of it, the chromosomes condense and they segregate. They first, as I said, undergo what we call a reductional division because it reduces the ploidy, meaning it reduces from two sets of chromosomes to one set of chromosomes, but they're still replicated. You still have two sisters here. And so there's a second division that separates the sisters to get to this 1N DNA content. So Gary, I think, mentioned that this crossover process is essential for the proper segregation of homologous chromosomes. It actually forms a physical link that holds the chromosomes together at this point and enables them to go away from each other during the division. And it seems very likely that the fact that this crossover process is mechanically required for the process of meiosis is a way that evolution has ensured that meiosis continues to be productive, because it's the crossovers that shuffle genetic material in a way that seems to be very advantageous for the organism. So there's a question as to whether reductional or equational division changes the ploidy. It's really the reductional division that changes the ploidy, meaning the number of chromosomes. But you know, we talk about N and C in chromosome biology. There's still two copies of each chromosome here, so they're 2C, but they're 1N because there's only one copy of each homolog. It's a little fuzzy because, as I said, the homologs are not purely identical anymore, or they're not purely different from each other. They've undergone recombinations, so the identity of sisters versus homologs is a little bit complicated. Okay, so this is not in the book, but it's something I think is quite important and very interesting. So there's a major change in chromosome organization as cells enter meiosis. So this is sort of a diagram of, you know, kind of an imaginary interphase chromosome, which, as I told you early on, are thought to be organized in these loop domains, and it's now thought that those loops are formed largely by cohesin complexes, which I also talked about on Wednesday, because they, in addition to forming loops, these cohesins also hold the sisters together in mitosis. What happens in meiosis is still very mysterious, but some of these cohesins get modified in ways that we don't fully understand, and they change their behavior dramatically and abruptly, so that meiotic chromosomes don't have this sort of globular territory in the nucleus kind of appearance. Instead, they're very long, and you can actually see them individually in the microscope, which is one reason that early cytologists were really fascinated by meiosis, because you can actually see a lot of what happens. In this case, the cohesins have sort of stacked together in a way that we, as I said, don't really understand, to form what we call the chromosome axis. And the loops of chromatin come off of the axis. It's likely that this is a dynamic structure, but it has this very rod-like appearance. And that's absolutely essential for everything that happens in meiosis. And I just mentioned here that meiotic prophase chromosomes are often described as condensed, but they're actually not condensed in the sense that we mean during mitosis until the end of meiotic prophase, when they shrink down and get ready to segregate. So during most of meiosis, you can see them as individuals, but they're still, the chromatin is not particularly condensed, it's just arranged in a different way. And what happens during early meiosis, and I won't have too much to say about this because we don't really understand it very well, but the chromosomes have to move around in the nucleus, and then somehow they recognize each other. And that part is still quite a mystery, how they do that. So they find their partner, and usually they pair up probably at particular regions. I'll talk about how telomeres often play a role in this, and they initiate meiotic recombination, and that leads to what's called synapsis, which is the assembly of this material called the synaptonemal complex between the chromosomes. Some people consider the axis to be part of the synaptonemal complex. I describe them as separate entities, just so that you're aware of the terminology. I won't talk about this today because this is definitely kind of at the cutting edge of research, something my lab is very interested in, but we now appreciate that this material between the chromosomes helps to regulate recombination. So it plays a role in holding the chromosomes together, and it also plays a role in regulating the number of crossovers along the chromosome. So how do the chromosomes find each other? As I said, a lot of that process is still mysterious, but one thing that happens in most organisms during meiosis is that the chromosomes form some kind of attachment to the nuclear envelope. So this shows some very early images of, in this case, a flatworm at the stage of meiosis that's sometimes called the bouquet, because it looks like a bouquet of flowers. And in this case, and in many cases, what happens is the telomeres, so the ends of the chromosomes, attach to the nuclear envelope. And we now know that they move actively by being attached to motors that are then moving along microtubules in the cytoplasm. And that sounds kind of confusing. I'll try to illustrate it a little bit here. So I don't know if I showed this image before. This is just a cartoon, again, of the nuclear envelope. I think I did show it because here's the nuclear pore complex, here's the lamina, which actually in some meiotic cells goes away. And what happens is that the chromosomes wind up attaching to these bridging complexes, which are sometimes called link complexes, for linker of nucleoskeleton and cytoskeleton. These are pairs of proteins, one of which is in the inner nuclear envelope and one is in the outer nuclear envelope, and they interact with each other in this luminal space. And there are different pairs of proteins that make up link complexes. The ones that act in meiosis seem to usually connect chromosomes to microtubules. So chromosome, here's the chromosome. In this case, it would be the chromosome end that links up with this sun domain protein and then this cache domain protein. And then this is a dynein motor. And what happens is that the microtubules in the cytoplasm act as tracks, and these complexes move along the nuclear envelope powered by dynein and probably other microtubule motors, and they move the chromosomes around. So this shows that in a different way, maybe a more useful way. So here are the telomeres, and they're forming attachments to these link complexes. And what often happens is that the link complexes cluster together because the highest density of microtubules is usually, if there is an active cytoplasmic microtubule organizing center, there's a high density of microtubules there, and they collect there to form this bouquet structure. I think that the literature really indicates that it's the movement that's important rather than the clustering, but people tend to fixate on the clustering. And then later, when the chromosomes are fully paired and synapsed, these go away and the chromosomes move around again, or they're no longer moving along the nuclear envelope. And I try to avoid using these terms, leptotene, zygotene, pachytene, et cetera, because I think they're just confusing, but you may find them in textbooks. Pachytene is defined as the stage where the chromosomes are fully paired and synapsed. Pachy means thick, and so they look really thick at this stage. And here they look thinner, or more thread-like, and that's where these names come from. Zygotene means coming together. So zygotene is the phase in which the chromosomes are actually pairing up. Okay, so all of this pairing and synapsis that, that completes by pachytene is essential because it enables the chromosomes to recombine with each other. And as I described in the introduction, those recombination events give rise to these structures that we call chiasmata. The singular is chiasma, the plural is chiasmata, that physically connect the chromosomes. And when they condense near the end of meiotic prophase, you can start to see these chiasmata between the chromosomes. Earlier, you can't see them because they're too close together and they have synaptonemal complex all in here. Sorry. Sometimes there can be more than one. In this case, it shows 3 chiasmata, and that's what's shown here. Often there's only one. So these chiasmata are the physical manifestation of crossovers, and they are essential, as I said, to hold the chromosomes together and enable them to segregate. And it's the combination of going through this recombination process that creates this cruciform structure and the sister chromatid cohesion that was initially set up between the sister chromatids, so the 2 blue ones here and the 2 red ones here, that holds this whole structure together until the first division. And at that point, each homologue attaches to one pole of the spindle, but the sisters stay together. So especially around the kinetochore region, the sisters stay glued together. So the sisters act as a single unit during the first division and only come apart during the second division. And I'll show that a little bit more clearly in a few slides. Okay, so I mentioned that recombination is tightly regulated and that the synaptonemal complex plays a role in that. I'm not going to say too much more about how that works. I know Gary talked a little bit about the different pathways of recombination, but I will just kind of illustrate that not all recombination events are crossovers. So early in meiosis, often right at meiotic entry, the chromosomes start undergoing these double strand breaks. And there are many of them. It's thought that they're about on the order of 10 to 15 breaks per chromosome in mouse, for example. We don't know in all organisms. So they undergo breaks, and in many organisms, those breaks help to kick off the assembly of the synaptonemal complex. And then at the point where the synaptonemal complex is more extended, there's some kind of decision made, and we don't fully understand how it works, but what happens is that a subset, so these breaks all start getting repaired through homologous recombination. And then a subset of them decides that they're going to become crossovers. And sometimes it's just one, and sometimes it's more than one. When there's more than one, they are spread out along the chromosomes. So there's a mechanism that we call crossover interference that prevents crossovers from happening near each other. So when you have multiple crossovers, they tend to be far apart on the chromosomes, and sometimes they're so far apart that you only get one. How this works is still a major controversial issue in the field, and so I'm definitely not going to explain it to you today, although I have plenty of ideas about how it works. But the upshot is that the probability of having a crossover at some region on the chromosome depends on the probability of having one somewhere else. So they're not independent. And so if you take more advanced genetics class, you will often do calculations of the crossover, the coefficient of interference, or there are other terms that are used, which basically is a measure of how strongly a crossover in one region inhibits the probability of there being one nearby. And it's different in different organisms. So you don't need to know all of that, but I do want you to be aware that not all recombination events during meiosis become crossovers, only a subset. And that's illustrated here. These are spreads of mouse spermatocytes undergoing meiosis. So this is an earlier stage and this is a later stage. And in this case, they're stained with a DNA dye in blue and antibodies to an axis protein in sort of pink or red. And in this case, the green is labeling a protein that's involved in the early stages of homologous recombination, a protein called RAD51, which you don't have to know. And in a mouse spermatocyte, people can count something like 250 individual recombination events that initiate, but then later, this protein marks the sites that are going to become crossovers, and there's typically only 1 or maybe 2 per chromosome. So there's about 10-fold more breaks and recombination events than there are crossovers. The ones that don't become crossovers get repaired in a way that gives what's called a gene conversion, so a local cutting and pasting of information from one parent to the other. So they do add to genetic diversity, but they don't create that cruciform structure, and they don't involve swapping of the entire segment of the chromosome. Okay, and the book talks a little bit about this, but it actually has what I consider to be a mistake in it, so I overwrote the legend here. It talks a little bit about the fact that so when you have genes that are on different chromosomes, those chromosomes are independent of each other. Whether you get one allele of a gene on one chromosome and another allele of a gene on another chromosome, those are independent events, and that's one of Mendel's principles. All the different traits that Mendel looked at, just by good fortune probably were unlinked to each other. They segregated independently of each other. But when genes are close together on a chromosome, the probability that there'll be a crossover between them is relatively low, and so they will tend to, the alleles of those genes that are together on the maternal chromosome or paternal chromosome will tend to segregate together. And that is known as genetic linkage. So people basically used the co-segregation of different traits in fruit flies and other organisms to make the first genetic maps, which is a map of physical, it's a map of genetic distance which correlates not perfectly, but with physical distance along the chromosome. So the farther apart something is physically along the DNA sequence, the more likely there is to be a recombination event between those two places. Okay. And then, so I started to talk about how the chromosomes actually segregate. And you might realize that something I said seems a little bit confusing, because we talked about mitosis, where the way that chromosomes segregate is that the cohesion between the sisters is dissolved, and that's what allows them to come apart. And it turns out that that has to happen in a more complex way during meiosis, because in order for these homologous chromosomes to let go of each other during the first meiotic division, you actually have to dissolve some of the cohesion that's holding them together here and here and here and here, but you have to retain cohesion between the sisters in this region around the kinetochore. And so that's kind of illustrated in this series of animated slides, which I think are helpful. So this would be what a chromosome looks like sort of at the end of meiotic prophase. It has a chiasma, a single chiasma. Here's the kinetochores. And what happens is that the synaptonemal complex goes away. So now there's not that glue holding the homologous chromosomes together. The only thing holding them together is the chiasma and the cohesion that was set up earlier, which is shown more clearly here. So here's the cohesins that are holding the sisters together, and there's the chiasma. And this structure is often called the bivalent, because it's going to orient in two, it's going to separate in two directions at the first division. So what happens is that some of the cohesion, as the spindle assembles, is released, and that enables the chromosomes to come apart. But the cohesion around the kinetochore region is protected until the second division, and that keeps the sisters together. And because this is super important, I summarize it in a couple of different ways. So this is just a summary of what I've just said, that in mitosis, all the cohesion is destroyed, as we talked about, by the action of separase at the only division. So it's removed all along the chromosome. But in meiosis, in the first division, you retain cohesion around the kinetochores. And people, this is actually kind of an old idea, that we thought that was mediated by a special glycine subunit called REC8, but it turns out that that's not enough. There's actually a protein called shugoshin, which is a Japanese word meaning guardian spirit, that is recruited to the kinetochore region and protects the cohesion from being dissolved at that division. So there are many ways that things can go wrong during meiosis because it's kind of complicated. And errors in meiosis, just like errors in mitosis, give rise to cells that have inappropriate numbers of chromosomes in them. And if those cells make it all the way to the end of meiosis and become functional gametes like sperm or eggs, they can get fertilized and give rise to aneuploid embryos. And that's what's illustrated here. So this, you can actually have missegregation, which is often in meiosis called non-disjunction, which I think is like a triple negative. You've got a not staying together. Anyway, not staying together. So they're supposed to come apart. Non-disjunction means they fail to separate. So this is an illustration of non-disjunction in the first division, but it can also happen in the second division. And by looking at the genetic complement of the gametes or the progeny, you can tell whether it happened in the first or the second division. Specifically, if you have an organism that gets an extra copy of the chromosome, you can tell whether it got the kinetochore region from both of the parents or just one, sorry, whether the extra copy is the same, has the same kinetochore or not, and that tells you whether it's segregated whether it missegregated at the first or second division. I made that very unclear, so sorry about that, but I'm not going to ask you to solve any problems where you have to do that. This just shows the karyotype of a male fetus that was probably diagnosed by amniocentesis, which is no longer going to be used very much, I think, but it used to be that if you were pregnant, that they would take some of the amniotic fluid, which contains cells from the fetus, and they would culture those cells until they were in mitosis and do spreads and then look at the chromosomes like this. And in this case, you can see that there are 2 copies of most chromosomes, but there's an extra copy of chromosome 21. And that is the most frequent chromosomal birth defect in humans is trisomy 21, which is Down syndrome. So the reason it's the most frequent is not necessarily because chromosome 21 is the most likely to missegregate during meiosis. In fact, I don't think it is, but most aneuploidies in humans are fatal. So they call it that they terminate a pregnancy often before a woman even knows she's pregnant. The reason that chromosome 21 is prevalent is because it is not fatal in embryonic development. Often chromosome 21 fetuses will survive and be born and will grow and be, even to adulthood. Because it's a small chromosome and having an extra copy, although it is deleterious and causes mental retardation and a lot of other effects on development, it is tolerated. It's not fatal. And there are a couple of other chromosomes, 13 and 18, in which trisomy can survive to birth. Monosomy, in which you're missing a chromosome, is not tolerated at all in humans except for the X chromosome. You can have an XO, which will be a female. It's Turner syndrome. Sex chromosome aneuploidies are actually not that uncommon. The X and Y chromosomes, particularly in the male, have special ways of segregating that are somewhat error prone. And so getting like XXY or XYY is not all that uncommon. XXY is called Klinefelter syndrome, and it's a very mild developmental problem.
I have a question about trisomy 18.
Does it have an equal likelihood at time of birth? I think trisomy 18 is often fatal during fetal development, but it can survive, but it is fatal after birth, unfortunately. So we do not understand why, but human meiosis is particularly error prone. This is a very often cited review. It's now almost 20 years old that talks about the prevalence of meiotic non-disjunction and how it arises, or which is still kind of mysterious. One thing that is mysterious is that there's a strong sex skew in where it happens. And it's a little bit weird because so a human female, although a woman is born with many, many oocytes, only one of those oocytes actually completes meiosis during each menstrual cycle. And so a typical woman will have about 450 menstrual cycles in her lifetime and produce 450 mature eggs. And each of those eggs is kind of a huge investment, they're big cells. Conversely, a human male produces about 50 million mature sperm per milliliter of ejaculate. So you can calculate, I guess, what that means in terms of lifetime production of sperm. I'll just note that the sperm counts in humans have dropped about 59% in the last 70 years, or 80 years, I guess, for reasons that are not well understood. And so would you guess that this process or this process is more error prone? Anyone want to answer on the chat? Yeah, it would make sense for sperm to be a lot more error prone because like these are, yeah, everyone thinks it's males, sperm, 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 sperm. In fact, female meiosis is much more error prone. And when people have traced like when you look at aneuploid fetuses and you look at where whether the non-disjunction happened in paternal meiosis 1 or 2 or maternal meiosis 1 or 2, the vast majority, for most chromosomes, happen in the mother. And we don't really know why. We don't really know why that is. We do know that during spermatogenesis, there are stronger checkpoints that prevent missegregation than in the female. That errors in meiosis tend to lead to dead sperm that don't undergo fertilization, but the oocytes that have missegregated chromosomes somehow survive and get fertilized. And we also know, for reason, although we don't know why, that there's a very strong maternal age effect. So you're probably familiar with this. There's actually, the curve is actually more U-shaped than was appreciated in 2001, in that very young mothers, like teenagers, actually have a higher incidence of non-disjunction. But as women age, the prevalence of aneuploid eggs and aneuploid fetuses goes up dramatically, in almost like an exponential way. So that sort of defies easy explanation. If it was just some sort of, I mean, people talk about the rotting egg hypothesis, that the eggs are just kind of going bad over time, but you would expect that to be more of a linear process or even an asymptotic process, not this very strong kind of acceleration as women get into their late 30s and early 40s.
Sorry, Abby, so I think someone had an interesting question early on where they were like sorry, I know it was a little bit further back, but they were asking about whether you would expect dosage compensation to occur in the case of trisomies.
Yeah, that's a really excellent question. Yeah, that's a whole nother subject. So yes, when you have sex chromosome trisomies, they do undergo dosage compensation. If you're triplo X, for example, that 2 of the X's get silent in every nucleus. And so triplo X is, I think, viable. So the way dosage compensation works in humans can deal with extra X chromosomes. Does that answer the question? I'm happy to talk at office hours about that in more detail. Having sex chromosome aneuploidy, though, does cause problems in meiosis because you have unpaired chromosomes, and there are meiotic checkpoints that can detect unpaired chromosomes and tend to kill the cell. So most aneuploidies cause infertility in both males and females due to a failure in chromosome pairing. We mentioned that chromosomes interact with the nuclear envelope to pair with their homologs. So does the nuclear envelope still break down during prophase 1? So no, the nuclear envelope is present until the very end of prophase 1. That's one way in which it's not a real prophase. It's more like a G2 phase. But it does break down it breaks down just like in mitosis as the spindle is assembling. It breaks down to enable the chromosomes to interact with the spindle. good question. Okay. So this is part of why, this doesn't answer the question of why there's this strong maternal age effect on non-disjunction, but it is a good reminder that most of the stuff that I talked about, all the pairing and synapses and recombination that happen in a woman, so all of her oocytes, all of that stuff happened when she was in utero. So when a female fetus is developing in the mother, that is when all that meiosis stuff happens. And then the eggs or the oocytes all arrest at the stage that people call a dictyate arrest. The chromosomes are partially condensed. Again, they're held together by chiasmata and cohesion. And it's only when hormonal signals initiate at puberty and you start maturing oocytes each cycle that the divisions happen. And so there are often decades between creating this bivalent structure and the segregation that has to happen. And somehow it's thought that this is not a completely stable structure. So there's a lot of theories about why that is. A lot of people favor the idea that cohesion may gradually decline and cause these chromosomes to let go of each other. In addition, there's evidence that as women approach menopause and undergo some hormonal changes, that their ability to form a good oocyte spindle seems to decline for reasons that we don't really know. So it's a very active area of research, but we don't really know exactly why. Oh, and I mentioned this earlier, that for reasons that are not clear, meiotic checkpoints that are sort of the safeguards against non-disjunction seem to be stronger in the male than in the female, which does not seem to make sense, but that's just the way it is. Just because I showed that kind of scary graph, especially for all the women in the audience, I thought I would also show this rather alarming graph of sperm decline. And I alluded to this in a previous slide, that the number of sperm per milliliter has actually dropped by something like 59% since the early 20th century. And not only the total number, but also the quality of sperm as measured by the percentage of sperm that are swimming properly and morphologically normal has also declined. We don't really know why. It's assumed that a lot of the chemicals in our environment, that many of which actually mimic reproductive hormones, may be contributing to this, and also pollution and other kinds of toxins in the environment. It's actually a study of meiosis that led to the whole backlash against a lot of the plastics. So you basically can't buy a plastic water bottle that doesn't say BPA free on it. And BPA, bisphenol A, is a chemical that is used in the manufacture of plastics. It keeps plastics kind of liquidy so that you can form them. And it was discovered actually totally by accident that bisphenol A disrupts meiotic chromosome segregation. This is a study in mouse. And what happened in this lab at Case Western was that they got a new custodial service, and the custodians weren't properly trained in how to clean the mouse cages. And so they started using this new, like a detergent that you shouldn't use on plastic, a harsh alkaline detergent. And that plus hot water wound up extracting BPA from the water bottles. And the researchers noticed they were studying spindle formation in oocytes from these mice. And normally this is like a wild type control, but they started seeing that their control mice had very bad spindles. And they eventually traced that to this new detergent that was being used to wash the water bottles, leaching the BPA out of the plastic. And we're able to reproduce this effect by giving the mice BPA. And that was the point at which like BPA-free plastic became like everywhere. Canada banned like drinking bottles and baby bottles containing BPA. But of course, they still use other plasticizing agents, many of which may also have similar effects that just haven't been documented. So we have a few minutes. Were there other questions? I don't think so. I'm going to just show you a couple of slides just from like, from my lab, so you can see how we look at meiosis. So we study meiosis in these nematodes and C. elegans. And one reason that they're great for studying meiosis is that the whole process happens in these tube-like organs, the reproductive organs. In this case, they're not, they make both sperm and oocytes, so they're ovotestes. And we can, and so this is just a cartoon showing that there's a pool, and this is typical of other animal reproductive tissues. There's typically a pool of germline stem cells. So those are cells that are maintained in a proliferative state by some kind of signal. In the worm, it's a signal that comes from a cell that's like right here at the distal tip of each part of the gonad. So it exerts control over this population of cells, but then when they move far enough away from that inhibitory signal, they enter meiosis. They do all the cool stuff that we study, the chromosome reorganization, pairing, and synapsis. That all happens within a few hours in C. elegans, and the chromosomes remain in this paired state called packaging for a long time, and then they condense and get ready to segregate, and they segregate. These will become oocytes. They actually make sperm first, and they store the sperm here, and as these cells come through, they get fertilized in here, and that triggers them to go through the meiotic divisions, and then they actually start developing internally. And this is what it looks like kind of in real life. This is a micrograph of just like one gonad from the from a C. elegans, from a worm. And we can actually look at chromosome pairing. In this case, we're using in situ hybridization to look at whether the two copies of each chromosome are together or apart. And so up here, they're apart. You can see two dots in each nucleus. And then this is about where they enter meiosis, and we can actually see that those dots can get fused together, and then they remain fused for this extended period. And eventually the chromosomes condense. And so at this point, they're just little tiny, like Tic Tacs. And we can see that they are held together by chiasmata. And when there are errors in this process, then so there's only 6 pairs of chromosomes in C. elegans. So there's 6 of these little bivalents. But if there are errors in meiosis, then we see up to 12 univalent chromosomes at this stage. So I don't know if seeing it in real life is actually helpful or not, but that's what we look at in my lab. So any questions? There was a question about what happens during dictated rest. Basically, this would be like, this is basically the stage at which human oocytes arrest. They kind of hang out in this stage where there's no more synaptonemal complex, but the chromosomes are held together. The homologous chromosomes are held together because they underwent recombination. In a worm, this stage lasts, a few hours max. But in a human, it can last from, from fetal development all the way up to when a woman ovulates. So that can be up to, 45, 50 years between all this stuff that happens to set up the bivalent structure and the actual segregation. And so somehow this is not 100% stable, which maybe isn't too surprising, and it can fall apart during that process, but we don't know exactly why.